Up to 51%. Like, that is just a staggering metric to start with. Up to half of your incredibly expensive exosome therapy might be, vanishing before it ever even reaches the patient.
Yeah, literally just adhering to the walls of the plastic tube you're holding.
Right. And you could be executing every single step of your protocol perfectly, but half of your active biological material is just gone.
It's wild. It really underscores the hidden variables in this field, I think.
Hmm.
We spend so much time focusing on the isolation, the
Yeah, the
characterization.
therapeutic potential.
Exactly, the potential of these lipid-bound nanocarriers. But we often completely overlook the perilous logistics of simply getting them out of a lyophilized state and into a functional liquid suspension.
Which is exactly our mission for today's deep dive. Welcome in, everyone. The mission today is to explore a highly specific, but honestly critical process. We're looking at the exact safety precautions required when reconstituting lyophilized or freeze-dried exosome powders.
Specifically focusing on the absolute mandate to use preservative-free normal saline. Right, because whether you are, working in regenerative medicine, running aesthetic therapies, or just running assays in a research lab, mishandling this reconstitution process has stakes.
Yeah.
I mean, we're talking contamination, severe patient harm, or, like we just said, losing half your expensive exosomes to a plastic tube.
And if you picture the setting for this, it's not a casual environment. You're in a high-tech, sterile clean room.
Sure.
And you have these vesicles, right? They're currently suspended in this highly stable glass-like matrix. Lyophilization basically preserves them by turning them into this powder, often using cryoprotectants like trehalose.
But waking them back up, that's where the danger is. It introduces immense chemical and environmental risks. Okay, let's unpack this.
Yeah, let's do it. So if we begin at the very first step, the environment and the liquid being used to wake the exosomes up, this is actually the most vulnerable moment for contamination.
Because it's the moment you transition them from that protected static state back into a dynamic environment.
Exactly. Now, the protocol overwhelmingly dictates reconstitution using an isotonic solution. So that's primarily preservative-free normal saline, 0.9% sodium chloride, or 1-xPBS.
And the choice of that specific diluent isn't just arbitrary, right? It's about osmotic safety.
Right, it's crucial. Exosomes are essentially nanoscale lipid bilayers enclosing an aqueous core. The sources give a strict warning here. If you were to reconstitute them with plain, sterile water...
Which people might just grab out of habit.
Totally. But sterile water intravenously without adding a solute is hypotonic. So the rapid influx of water into the exosome to, equalize that osmotic gradient, it would swell the vesicle past its critical lytic volume.
Oh, wow. So it just bursts.
It ruptures the membrane entirely, yeah. The osmotic imbalance causes hemolysis, basically red blood cells bursting, and the same principle applies to these delicate vesicles.
That makes sense. But let's look at the preservative-free part of the mandate, because honestly, that feels counterintuitive. I mean, it's like opening the door to a highly secure vault, but there's no security guard inside to fight off intruders. Once the door's open, anything can get in.
That's a great way to picture it.
Right. So if preservative-free saline is so risky for contamination, why not just use the bacteriostatic version and save ourselves the headache? In a busy clinic, throwing away a whole vial after one use feels incredibly inefficient.
This raises an important question because, the intuitive leap is exactly what you just said. Adding an antimicrobial agent should increase patient safety, right?
Yeah, to prevent contamination.
But we have to look at the specific chemistry of that preservative. Bacteriostatic saline typically contains 0.9% benzyl alcohol. And benzyl alcohol is an aromatic alcohol. When it contacts delicate tissues or lipid vesicles like exosomes, it doesn't just sit there inertly. It actually partitions into the lipid bilayer. It disrupts the hydrophobic interactions between the lipid tails.
Wait, so it actively attacks the structure?
Exactly. It increases the membrane fluidity to the point where the vesicle becomes highly permeable. It leaks its cargo, or it just completely disintegrates.
So by trying to prevent bacteria, you're effectively trading microbial security for the complete destruction of your actual therapy.
You're chemically shredding the exosomes. So yeah, preservative-free saline is completely non-negotiable.
Wow.
But because it lacks preservatives, it is strictly single-use. The CDC safe injection practices are very clear on this.
Right. The sources outline those CDC protocols pretty rigidly. You have to use a brand new sterile syringe and needle for every single application.
And you can never re-enter a vial with a used needle. Never. Any unused portion has to be immediately discarded.
No saving it for later. And we're also dealing with biosafety levels here, depending on the source of the exosomes, right?
Yeah, depending on the source, you need BSL-1 or BSL-2 precautions. For instance, if you're dealing with human-derived cells, that requires BSL-2. So you're using protective screens, gloves, biological safety cabinets, the whole 9 yards to prevent cross-contamination.
Okay, so we've navigated the first major hurdle. We've got our sterile field. We're using isotonic preservative-free saline so we don't chemically shred or burst the exosomes. Now we actually have to mix it.
Right, the physical act of mixing. And it turns out human error right here can easily destroy the very exosomes you're trying to save.
Because the physical forces are just as destructive as the chemical ones?
Completely. So the protocol requires starting with an ice-cold buffer or saline. Usually the volume ranges from 25 microliters to 1 milliliter, just depending on the concentration you need.
Why ice cold, though?
Well, keeping it ice cold minimizes thermal kinetic energy while that cryoprotectant matrix, the trehalase we mentioned, dissolves. It stabilizes the exosome's structural integrity during rehydration.
Okay, so you add the ice cold saline, then what?
Then you do nothing.
Nothing. You just wait.
You just wait. The sources specify an incubation pause. Before mixing, the solution must sit entirely undisturbed at room temperature for 5 to 10 minutes.
Just to let the pellet dissolve completely on its own.
Exactly. If you rush it, you subject structurally compromised exosomes to massive mechanical stress.
It sounds like we aren't shaking a cocktail here. We are folding a highly delicate souffle. The physical forces are just as dangerous as the biological contaminants.
If we connect this to the bigger picture, you have to remember the fragility of that lipid bilayer. An exosome is roughly 30 to 150 nanometers in diameter, so we're talking incredibly small, incredibly fragile.
So shaking a vial vigorously, which is what most clinicians do instinctively with a powder, is just completely off the table.
Oh, absolutely off the table. If you shake it, you create massive shearing forces. And on a nanoscale, those shear forces physically tear the exosome apart.
Wow. Okay, so how do we actually mix it then after the pause?
The required technique is gentle pipetting. You draw the solution up and down very smoothly about 10 to 15 times for dispersion. But the major precaution here is that you absolutely must avoid creating bubbles.
Bubbles. I mean, a bubble seems harmless. It's just a pocket of air.
You'd think so, but foaming causes exosome aggregation and severe damage.
Wait, really? Just a bubble?
Yeah, because a bubble creates an air-liquid interface. And the surface tension at that boundary is incredibly intense on a nanoscale. When the exosome touches that interface, it literally unfolds the vesicle.
Oh, see.
And when the bubble pops, the hydrodynamic shockwave exerts this instantaneous physical force. The exosomes either rupture violently or their hydrophobic cores get exposed and they clump together into these non-functional masses.
Just to escape the water. That makes total sense. So gentle pipetting only? But what if there are still clumps? Is vortexing allowed at all?
If vortexing is absolutely necessary for clumps, it must be strictly limited. The sources cap it at a maximum of 60 seconds.
Just a minute.
Right, any longer. And those continuous shearing forces will shred the lipid bilayers.
Okay, and I saw something about centrifugation in the sources too. Isn't that a huge physical force?
It is, but it's a brief, low-speed centrifugation. We're talking 1,000 to 3,000 times gravity.
Oh, okay, so just a quick spin.
Exactly. It's just required to pull all the liquid down to the bottom of the tube, to prevent any material loss from droplets clinging to the sides during handling.
Okay, so we've been perfectly gentle. No bubbles, limited vortexing, quick spin. We have our perfectly reconstituted exosome solution sitting in a standard plastic tube. And this brings us back to that terrifying metric from the intro.
Right. The hidden danger waiting in the very tube you are holding.
Here's where it gets really interesting. Because you maintained a completely sterile field, you mixed it perfectly, and yet the therapy is literally vanishing. Let's talk about the Yevtushenko study.
Yes, the POS1 study. It is fascinating data. They used exosomes isolated from human colon cancer cells, HT29 cells, and they stored them in ordinary, everyday polypropylene tubes at +4 degrees Celsius.
So standard fridge temp.
Right. And the concentrations were substantial, like 0.5 to 2.1 times 10 to the 10th particles per milliliter.
Billions of particles. And what happened after 48 hours?
Up to 51% of the exosomes were simply lost.
Over half. In 2 days. Why does it happen? Are they degrading?
No, they aren't degrading. They're literally sticking or adsorbing to the plastic walls of the tube. Polypropylene is inherently hydrophobic, and exosomes have localized hydrophobic lipid domains. When they bump into the wall, they bind to it permanently.
Wow. And the study highlighted that it doesn't happen evenly to all the exosomes, right?
Exactly. It's a diffusion-controlled absorption. And because of how diffusion works, it disproportionately claims the smaller exosomes.
So if the smaller exosomes are disappearing faster because they diffuse and hit the walls quicker, are we actually changing the therapeutic profile of the medicine just by letting it sit in a plastic tube?
What's fascinating here is that, yes, absolutely. The biological activity could be fundamentally altered by this passive loss. You're losing the smaller population, which carries specific molecular signatures, so you're altering the size distribution of the entire sample.
You're basically delivering a completely different therapy than what you started with.
You really are.
So how do we stop this? What are the safety countermeasures here?
The primary defense is speed. To prevent this massive loss, the reconstituted solution must be used immediately, ideally within 2 hours.
Okay, so reconstitute and inject or apply right away. But what if you can't? What if storage is required for an experiment?
If storage is absolutely required, you have to use specialized containers, specifically Eppendorf protein low-bind tubes.
The low-bind tubes.
Yeah, they have a modified surface that prevents the hydrophobic domains of the exosomes from anchoring to the plastic. Alternatively, if you don't have those, you can block standard tubes with bovine serum albumin, or BSA.
So the albumin coats the plastic first so the exosomes have nowhere to stick.
Exactly. It occupies all the binding sites.
That's incredibly clever. And what about long-term storage?
For long term, you need to store small aliquots at -20 degrees Celsius, which is good for up to 1 month, or at -80 degrees Celsius for up to 6 months.
But freezing has its own risks, right?
It does, which is why you must avoid more than 2 freeze-thaw cycles. Every time you freeze and thaw, you risk protein denaturation and structural damage from ice crystals.
So what does this all mean? We've covered so much ground here. Let me recap the triad of safety for exosome reconstitution for you, the listener.
Please do.
First, we have strict aseptic protocols. You absolutely must use preservative-free, single-use saline to prevent chemical shredding while maintaining a perfectly sterile BSL-1 or BSL-2 field. Second, incredibly gentle physical mixing. We're talking ice-cold buffers, a mandatory 5 to 10-minute pause, gentle pipetting, and strictly avoiding bubbles and excessive vortexing to prevent shear forces from ripping the exosomes apart.
Spot on.
And finally, managing the container itself. Using the solution immediately or relying on specialized low-bind storage and controlled freezing to prevent your vital therapeutics from just vanishing into cheap plastic.
It's a very intense set of protocols, and, I think it leaves us with a really provocative thought to mull over.
What's that?
Well, think about it. We are currently engineering incredible, cutting-edge nanocarriers to deliver revolutionary therapies. Billions of dollars are going into this.
Yeah, it's the frontier of medicine.
Yet their ultimate clinical success might be entirely dependent on outsmarting the static cling of a cheap, everyday plastic tube.
Wow, that's a great point.
Right. If nanotechnology is the future of medicine, the material science for the containers holding them simply must evolve just as fast. We can't put 21st century medicine in 20th century plastic.
That is perfectly said. It really puts the whole process into perspective. Thank you for joining us on this deep dive, everyone. We highly encourage you to take these rigorous standards and apply them to your own clinical or research practices. Don't let half your therapy disappear before you even use it.
Thanks for tuning in.
Catch you next time.